Hello, and thank you for listening to my talk. I wanted to thank Elucidata for this opportunity to tell you a little bit about some research that we've been working on at the University of Florida in collaboration with Moffitt Cancer Center. The title of my talk is Digging into the Cancer Metabolome with Liquid Chromatography High-Resolution Mass Spectrometry, specifically looking at the changes in lipids related to breast cancer cell lines, specifically in the reduction in the pH. Acidosis in cancer is a key area of study. We know that malignant tumors exhibit an altered metabolism. Part of that metabolism, of course, related to how they utilize glucose. The increased glucose metabolism leads to an acidic microenvironment around tumors. It's also known that cancer cells have to use this environment and adapt to it to continue to grow and proliferate. That adaptation leads to development of lipid droplets or adiposomes. This is also known as adiposomogenesis and the process of lipid droplet formation. They contain in their core primarily triglycerides and cholesterol esters, and in their shell, creating the sphere, has polar lipids, typically phospholipids like phosphatidylcholine, as well as perilipid proteins. We know that they play a key role in energy storage, production of energy, and membrane production for lipids, as well as in lipid signaling processes. We also know that they've been observed in many different metabolic diseases such as diabetes, fatty liver disease, and cancer. Lipidomics is the detailed study of lipids, typically using mass spectrometry, often coupled with chromatography. And we know there's well over 1,000 bioactive lipids to date, with many functional consequences. They're involved in numerous disease states, so they have biomarker potential. They're also involved in signaling, as I mentioned before, and they're ubiquitous in many samples, relatively easy to ionize by mass spectrometry, and often highly concentrated. We also know that they're very difficult to separate all lipid species because of the many different isomers and isoforms. One example of that is looking at glycerolipids. Glycerolipids start with glycerol, and these would be triglycerides. You can lead to monoacylglycerides, or MGs, and also diglycerides, and then finally triglycerides, where we have 3 fatty acyl chains attached to the glycerol backbone. Those can also be further modified to make plasmanyl triglycerides, which would be 1 ether-linked fatty acyl chain. If you go through all the potential combinations of this, there are over 10,000 potential combinations using 39 common fatty acyl chains throughout this whole process. That's a lot of different potential species present in a given sample. All of those may not be present, but all of those have the potential to be present under a given situation. So why are we interested in lipids in cancer? Well, we noticed in this collaborative project that we can form adiposomes when we grow cells under acidic conditions. And this graph shows you the amount of lipid droplet formation, an increase in MCF7, MD8, and T47D cell lines as we lower the pH from 7.4 to 6.5, showing a large increase in the amount of lipid droplet formation. We can also look at key staining of cells to understand whether or not these are true lipid droplets around cells. And we can look at the average lipid density increases and changes with different cell lines. Using Nile red, we can see that we have an increase in the amount of lipids present in droplets at 6.5. We can also stain for perilipid proteins, specifically PLIN2, and we can see an increase around the cell environment. And part of that also involves, when you merge this together, you get a much clearer picture. So you can see that we definitely get a clear increase in the amount of lipids when we lower the pH. We also know we can alter the extracellular intracellular environment using sodium gluconate, and this is demonstrated with MCF7 cells. Sodium gluconate reduces the chlorine gradient, and it often leads and will lead to a higher steady state intracellular pH by changing out the amount of chlorine in the cell environment. We don't alter the amount of adiposome accumulation. So we still see this accumulation. So we know that it's just not related to the cytoplasmic pH. Next, we looked at P13 kinase signaling. So we know, and specifically we looked at OGR1 and found that OGR1 is highly expressed in MCF7 and T47D cell lines. OGR1 is an acid sensing receptor. We then targeted the specific protein by inhibiting it using a key inhibitor and comparing it to a separate acid sensor TDAG8. You'll notice in the figure that when we inhibit OGR1, we see a clear reduction in the amount of lipid droplet formation that's produced. And when we inhibit TD8, we don't see that clear lipid droplet reduction. We can further use a knockout model where we can knock out OGR1, and we can run at different pHs, and we look at both a neutral as well as an acidic pH. And under acidic conditions, we don't see this droplet formation when we knock out OGR1. Furthermore, we can alter the pH by increasing it. And when we increase the pH, we know that OGR1 is inactivated, but we can reactivate it using a drug called Ogerin, and we can get a dose response based on dose response and lipid accumulation. And if you see that on the bottom part of the figure, you'll see at 1 micromolar versus 5 versus 10, we see an accumulation of lipid droplets. So even when we turn off OGR1 by raising the pH, we can turn it back on using Ogerin and see this increase in lipid droplet formation. So overall, what we can observe from this is that by blocking OGR1, we can reduce lipid droplet formation, a potential treatment product. So we also know that the G protein coupled receptors, such as OGR1, they activate P13 kinases and the phosphorylation of AKT. And we can see this we can confirm this by evaluating the phospho-AKT. At pH 6.5, we see a huge increase in the amount of AKT present. And we can also use an inhibitor of this phosphorylation process, both at the PI3 kinase level and the PAKT level. That's what the top and the bottom figures. And at 6.5, when we inhibit this, we can reduce the amount of lipid formation. So we can block both of these pathways and reduce the amount of lipid droplet formation. Finally, we can also determine whether or not this adiposome accumulation is dependent on autophagy. And we evaluated different cell lines, both MCF7 and T47D. I'm only showing you MCF7 in this cell line. And one of the questions we had was whether the acid-induced lipid droplet was indeed dependent on autophagy. And so MCF7 cell lines were treated with 2 different inhibitors, both chloroquine and bifilomycin. And we have again observed a significant reduction in lipid droplet formation using both inhibitors, which suggested a connected role of adiposome formation and autophagy. If you recall from before, we used the OGR1 knockout model to confirm the role of OGR1 in lipid droplet formation. And we used the same model to assess this acid-induced autophagy. TFEB is a master regulator of autophagy. And as you can see, we observe an increase in TFEB when the pH is reduced to 6.5. In the OGR1 knockout model, we observed lower levels of TFEB as well. And this helps to confirm that indeed the acid-induced autophagy is triggered by OGR1. With this information, we then wanted to explore the role of small molecules as carbon source for lipid droplets. So we set out to measure key species that would relate and be involved in lipid metabolism. So we evaluated the fate of 5 different precursors using isotopic labeling, primarily carbon 13. And we used leucine, glucose, lactate, acetate, and glutamine. Each precursor was provided in the cell media of each breast cancer cell line, left there for 72 hours to incubate, and then removed, and we isolated the lipid droplets by centrifugation. And then extracted the lipid droplets, again, using chloroform methanol just to separate out any polar and nonpolar lipid species. We then used high mass liquid chromatography coupled to high resolution mass spectrometry, as well as tandem mass spectrometry to identify lipids and to help classify which was in which species. Key to this was a collaboration with Elucidata to help us understand the fate of lipid labeling. And we used and collaborated with Elucidata using Poly and translated it for lipid labeling evaluate this complex species. And I'll show a slide in a little bit that shows you what it looks like. But to back up, the reason we chose leucine was to identify whether or not the breakdown of ketogenic amino acids from the autophagy process would lead to lipid storage that could then be utilized for future use within the cell. So as a part of the labeling, we indeed saw an increasing amount of labeling related to leucine. You can see from the figure at the bottom where leucine accounts for the second most amount of carbons in triglycerides, second only to glucose, which is well known. And in each cell line, we have a range of 14 to 16% carbon contribution from carbon 13 leucine. And we also confirmed that glucose was primarily present in the glycerol backbone, while leucine was observed primarily in the fatty acyl chain, suggesting a different process of accumulation. You can look at individual labeling across all the multiple classes of triglycerides detected, and we detected well over 50 different triglycerides in the sample sets. And we can see that MCF7 has a very large number of carbon contribution from leucine in the triglyceride content. A significant amount more than the other two cell lines involved. Part of what the complex part of this is what an example of a lipid spectrum that we would see and we would observe in a sample region. You can see this is summed across a specific range of triglycerides that were present. So each one would be somewhat separated based on chromatography. But if you just look at the sum of what we would observe, and this is a labeled study with leucine, you can see a very detailed mass spectrum that's very difficult for us to decipher. The only way we were really able to work with this type of data in this complex environment was through this collaboration where we did a picking program with Maven, looked at labeling with the precursors and used poly as a way to decipher that labeling. And really without this, we would not have been able to look at this very complex lipid process and really decipher what kind of lipids would be labeled with these different precursors. Also using Maven, we can also look at the total lipid content, those that were labeled or unlabeled in the process. And this just looks at the overall lipid content of adiposomes in different cell lines. You can see a drastic difference in different cell lines. In the MCF7, we have a large number of triglycerides representing almost 60% of the total content of MCF7, while in ZR75, the triglycerides only represented roughly 17%. And phospholipids, primarily phosphatidylcholine, represented the large portion of the other part of that adiposome. There's a lot more work that we have to do to look into this key part of lipid metabolism, but it shows the possibility using labeling as well as using high-resolution mass spectrometry coupled together with a key bioinformatics software provided by Elucidata and Poly that allows us to really understand the key differentiating factors in acidosis related to cancer biology. And with that, I'd like to thank you for your attention, and I'd like to thank the funding agencies for this, including the Bankhead Coley program from the state of Florida, the NIH, our center at the Southeast Center for Integrated Metabolomics at the University of Florida, and of course a collaboration with Elucidata. Most of this work was conducted in my lab by Iqbal Mahmood, who's here in the middle, and other people in my lab are key members in what we do in this work. And with that, I'd like to thank you very much, and we'll take any questions.